what did Evo2 do that was so, let's say, groundbreaking, and what is Evo2?
Right. So it's a fascinating project. And I feel like I was so lucky to be involved in this project. So Evo2 is really a groundbreaking AI model designed to understand and generate DNA sequences from all forms of life, right? From bacteria to humans. So think of it as a language model. We all are kind of becoming familiar with ChatGTP and how it can produce, respond to sentences, etc. So think about Evo2 as a language model for biology. So instead of predicting the next word in a sentence, it predicts the next DNA base pair in a genome, right? So this allows Evo2 to really analyze and design biological systems at a molecular level. And so it can help researchers predict, for example, how genetic changes or gene modifications might affect organisms, or it can even think about how we might generate new DNA sequences that could be used in synthetic biology or for drug treatments or for medications, etc. So really important in that sense, in that it's the first, DNA large language model at this scale.
Yes, and the thing that fascinated me quite a bit with this model, in some ways, it is like the LLMs that everyone's familiar with, especially ChatGPT, in that somehow the individual bases that make up DNA, the A, T, C, G, is common amongst like all species, all living life. But what was surprising to me was by training this model on such a diverse set of species. It wasn't just human genomes, but it was genomes from across the animal kingdom, that it was able to understand like how a genome is put together. That there's almost like a semantic layer that's above the bases that it was able
Right.
to understand, that's common amongst the kind of all the everything that's like, I guess, a lot of life within on Earth. Am I understanding that
That
correctly, or am I
that's
just making that up?
that's exactly what it is. And when we think about in the past how we've studied biology, we've studied these different levels of life forms. So C. elegans, yeast, to understand the basic fundamentals of life, cell cycle, things, DNA synthesis. And so that is kind of the concept behind Evo 2, is we really wanted to understand the evolutionary pieces of these basic fundamental forms of life, to understand how they fit together in this complex DNA sort of a dictionary of sequences. So, and just to, say a little bit more about that, because there were so many different forms of life included. Evo 2 was trained on 9.3 trillion DNA base pairs, right? Covering just a wide range of organisms, from bacteria to plants to animals. And so that's really where it had the power to learn these really important fundamental pieces of, or aspects of functions of life. So that was kind of what was unique about it.
Yeah, I guess what surprised me a lot, and I actually had some other comments from others on LinkedIn about this, was it wasn't trained on a huge amount of human genomes, which is, a lot of people assume that in order to understand what makes humans is you got to have a ton of genomes. So there's like the 100,000 genomes project in the UK. I think there's a million genomes project in either China, and I think the US has all of us as well. So there's a large amount of genomes being produced from that effort, and it's to understand sort of what makes us in biology and disease and so forth. But this was not done that way. This was done on so many species, so that it surprised me. It's almost like we have a this common layer. Did that surprise your team, or no? You kind
Well,
of expected that to.
that was very intentional. And here's something that so my team was really focused on: how do we think about the safety and the ethical aspects of this, right? And one of our big concerns was bias in the human reference genome, Right. So there's been a lot of research coming out showing the human reference genome is actually lacks diversity, right? It lacks a lot of um, um, sequences or genome aspects from Africa, from other areas of the world. It's very European Anglo-centric. And so we were very concerned that if we were, training Evo using this human reference genome, that we might learn these biases. We might learn very specific traits to this particular population. And so that was what was, I think, was really amazing about Evo2 is we were able to learn these functions, these gene functions, protein functions from these different species and actually see how we could pull this together to understand some aspects of the human genome. And in the Evo2 paper, we gave they provided, an example using a breast cancer oncogene. And how we could predict how modifications in that oncogene were really important or change gene function. So that is a perfect example of how we could learn these functions from plants, from bacteria, et cetera, to make insights into the human genome.